Single gene disorders occur when the DNA sequence for a specific protein is missing or altered. Tay Sachs disease is a metabolic disorder derived from a single gene mutation. Tay Sachs was collectively discovered in 1881 by Warren Tay and 1896 by Bernard Sachs. Although they were describing the same disease, it was not until 1930 that it was identified as a metabolic disorder, thus naming the disease Tay Sachs. Later, in the 1960s, GM2 ganglioside, the fatty acid responsible for Tay Sachs, was recognised and named. Tay Sachs is inherited in an autosomal recessive manner. This means that 2 copies of the faulty gene must be inherited for the disease to be present and that both parents must be carriers. Children of parents that are both carriers have a 50% chance of becoming heterozygous healthy carrier and a 25% chance to be either homozygous healthy or homozygous affected. Tay Sachs is a single gene mutation of the HexA gene, located on chromosome 15. Its position is 15q24.1. This means it's on the long arm of the chromosome at position 24.1. Beta-hexosaminidase A is an important enzyme found in the lysosome, which breaks down the fatty acid GM2 ganglioside. This enzyme requires the alpha and beta subunit in order to function correctly. The HexA gene is responsible for producing the alpha subunit, so a mutation to this gene is detrimental to the healthy functioning ability of the lysosome. A normal functioning lysosome allows the breakdown of the fatty acid GM2 ganglioside and for it to be disposed of. Carriers have enough of this enzyme to avoid symptoms. Without the proper functioning ability of this enzyme, the GM2 ganglioside fatty acid builds up in the brain and spinal cord, leading to cell death and the signs and symptoms of Tay Sachs disease. Depending on the severity of the mutations on the HexA gene will depend on the severity of the disease. Tay Sachs disease can be inherited in 3 different forms, and as seen from the slide, 1 in 250 people of the general population are at risk of being a carrier. The most severe is the infantile form of Tay Sachs. Symptoms are not shown until the infant is around 6 months of age, and at this time, development begins to become recessive in nature. There are less severe forms of Tay Sachs disease, juvenile Tay Sachs, which will show signs around 2 to 5 years of age, and late onset Tay Sachs, which typically appears around adolescence or later in life. All forms of Tay Sachs have no cure. The most common sign is the cherry red spot on the retina of the child's eye, as seen here, indicating excess storage of lipids. The child will eventually be unable to swallow or move at all, they will become blind, suffer from seizures and dementia, and be unable to breathe. The disease is very fast acting, and a child will die between the ages of 2 and 5 years, usually from respiratory failure. These photos show the rapid decline in children with the disease, with only 18 months between photos. There are a number of tests for diagnosis and screening single gene mutations, including Tay Sachs disease. Carrier tests can be done by enzyme assay, screening for beta-hexosaminidase A levels, or DNA tests screening for the specific mutation. Both are done by blood test. Pregnancy screening can be done by chorionic villi sampling, otherwise known as CVS, and is a procedure where some of the placenta is biopsied from the mother. This occurs around 11 weeks gestation. Amniocentesis, occurring around 16 weeks gestation, involves sampling the fluid from the amniotic sac. 2 testing protocols can be employed: fluorescent in situ hybridization, otherwise known as FISH, which uses fluorescence to detect abnormalities on chromosomes, or a polymerase chain reaction, otherwise known as PCR, which is used to amplify a specific DNA sequence. These methods are used to test for chromosomal abnormalities, including single gene mutations. Assisted reproductive technology, otherwise known as ART, can be used in families who wish to conceive but are aware of a genetic or chromosomal disorder within their family. In a couple who are both known carriers, a type of ART, such as in vitro fertilization, can be used in conjunction with a pre-implantation genetic diagnostic test to ensure the baby does not inherit both of the faulty genes. This type of ART involves a genetic profiling of the embryo outside of the womb before implantation. Currently, for late-onset Tay Sachs disease, there is research into an anti-malaria drug called PMT, which has the effect on the body of increased HexA production. Regarding infantile Tay Sachs, a recent discovery in Jacob sheep may allow further tests which are applicable to human infants, but until this time there is still no cure or treatment.